I am recording this one because there's a lot of material, and like we did with the hormones, there's a lot here because now we're going to be getting into the specialized parts of the blood, the formed elements we've already mentioned, red blood cells. But red blood cells are simple. White blood cells are not. There are two main groups of white blood cells. The first are called the granulocytes. The granulocytes. Because when you look at them under the scope, it looks like they're full of big grains. The other group are called the agranulocytes, lacking the grains. And we call them white blood cells in total because they're not pigmented with hemoglobin. Their size is very variable. Generally, they will be obvious when looking at a blood sample. The normal range is a minimum of 3,500 cells per microliter to a maximum of 10,500 cells per microliter. And as you can see, that is quite a bit lower than the millions of red blood cells, isn't it? Y'all saw that when you looked at the slide. It was kind of obvious. And all of these are involved in the immune response. One of the things a doctor will very often order is what they call a WBC test, a whole blood count. And what they're doing, they're getting the number of total blood cells, they're getting the total of red and white, and they're looking at, is it high, is it low, or is it normal? If it is low, it is called leukopenia. Leukopenia. Leukopenia will often result from a viral infection. And that can be, measles, mumps, flu, polio, HIV. There's a lot of different ones. It may also be the result of heavy metal poisoning, lead, mercury, arsenic. The reverse of that is called leukocytosis. Leukocytosis. And this is a high white blood cell count. Leukocytosis is almost always due to an acute infection. Bacterial, viral. It doesn't have to be systemic. Doesn't mean you're septic or going into septic shock. It just means that somewhere in your body there is something growing there that shouldn't be, and your body is trying to get rid of it. It can also result from dehydration or overexercise. Is everybody cool with that? Now, just as erythrocyte is the proper name for red blood cell, leukocyte is the proper name for the white blood cells. And that's regardless of whether it's a granulocyte or an agranulocyte, you just need to know leukocyte is the general term. I am not going to be overly picky about whether or not you use leukocyte or white blood cell, or if you just want to use the abbreviation WBC, because that's a fairly standard thing. I use that because as you go on up in the higher levels of courses, you're going to encounter the term, and you make sure that you understand sometimes using the proper term is better. That in white blood cells, what you're going to want is really to focus on what exact group of white blood cells does it fall into. There are 5 different groups spread amongst the 2 classes, and each of those has a very distinct function, and we'll be getting into that. So everybody with me?
Mhm.
Okay, so back to the granulocytes. The granulocytes are called that because, as I said, it looks like they've got grains in them. Wright's stain, W-R-I-G-H-T, proper name, is named after the person who developed it. It's a very, very common stain that you put on slides of blood, because back in the old days, it's how they used to diagnose a lot of things. I have no idea if they are still doing Wright's stain or things like that in a clinical diagnostic sense, but it's helpful to understand that's where the classification comes from. And there are 3 different classes of granulocytes. The first, well, a couple other things about granulocytes. They are very short-lived, typically only about 12 hours. So as you might imagine, when you have high numbers of these, it's almost always indicative of an infection. First group up are called neutrophils. These are the tripwire or the sentries or the first line. In other words, whenever the bacterium or virus or whatever is detected, the neutrophils are the first on the scene. They're the first ones to attack the invader. They kill invaders with a burst of toxic chemicals. Your book calls it a respiratory burst. I have no idea why. But it's just really, really nasty stuff, like the equivalent of bleach or hydrogen peroxide. Y'all have ever poured hydrogen peroxide? You see how it bubbles and reacts. They're very strong oxidizers. So they literally just dissolve whatever they come into contact with. The neutrophils are also full of lysosomes. And what they do is they break it down.
Lysosomes, you said?
Lysosomes. That's an organelle that's just a sac full of digestive enzymes. Anytime you have a variant number of neutrophils, it can be indicative of something like an autoimmune disorder. So any questions about neutrophils? All right. Next up, you've got eosinophils. Eosinophils also attack invaders. But what's interesting is the invaders they attack are neither viral nor bacterial. Instead, it's typically something like a worm or protozoa, something like malaria, for example. Roundworms, tapeworms. So even though those are macro scale invaders, your immune system can and will attack them, and it can and will defeat them. As part of that process, they also help to regulate the inflammatory response. The way they attack is they release cytotoxic proteins that literally break down and digest the parasite. They can both release and clean up the biochemicals that initiate an immune or inflammatory response, things like histamines, prostaglandins. So any questions on eosinophils? All right, the last of the granulocytes are called basophils. These are the rarest white blood cells in existence, less than 1% of them. And these play a major role in the inflammatory response and allergic reactions. They're the ones that are chock full of things like histamine, heparin, which is a biochemical that's involved in clotting. And these are the ones that release those chemicals when they encounter a foreign substance in the bloodstream. So if you pick up poison ivy or scrape it, there's a chemical called urushiol, and it's a oily-like substance. It gets absorbed from the skin, gets into the bloodstream. The basophils come into contact with this and they go crazy. They start releasing massive amounts of histamine. And that's why you get the rash, the swelling, the itching, and so forth. They're also involved in response to some worms, parasitic worms. They also release cytokines. I think did I talk to you all about the 1918 flu and the cytokine storm and all that?
That was the Spanish flu, correct?
Yeah.
Yeah.
Okay. The main thing about basophils to remember is they're essentially chemical releasers. They're the ones that mediate that response by the release of chemicals. I learn something from you all every day. Morgan's over here, she's written like 4 lines, and I'm like, is she taking notes? And then I look over here and she's highlighting everything in the book. I do it the old-fashioned way, and I use pencil and paper. So everybody comfortable with granulocytes? You will need to know whether a white blood cell is a granulocyte or an agranulocyte. You will need to know what it arises from. In other words, what type of stem cell gives rise to it. And you'll need to know the function. And I'm sorry, I know that's a lot of material, but as you see, there's a lot more than just a white blood cell, isn't there? They're involved, all of them are involved in the immune response. So you can always say they're involved in the immune response as a primary function. But how they're involved, when they're involved, those are the kinds of things you're definitely going to need to know. So what's the first one that always responds?
The
Neutrophil.
Oh,
Neutrophil. Which one's the chemical messenger?
eosinophils.
Basophil. Do I need to go out and get my chocolate-covered espresso beans and pass them out?
I've had those before.
Huh?
I've had those before.
Did I ever tell you about the time I was teaching a 3-hour lecture and lab combined on a Friday afternoon?
That sounds terrible.
It was. We didn't start until 3 and we got out at 6. And after a half hour, everybody, including me, was gone. So I turned to my teaching assistant, handed her my office keys, and said, Kristen, I want you to go up and get this box that's out, or the tin that's out of my right-hand drawer. She said, the secret drawer you never unlock? And I said, yes. This was a rumor amongst the students. I was wondering what I had in there, like cocaine or something. She comes down, she's got this big grin and pulls out my tin of chocolate-covered espresso beans. So I just set it on the first desk and said, take as many as you want and pass it on. By the time it went around for the third pass through the class, we were all excited about biochemistry. All right, is everybody awake now? Okay. I can't see Ava because of the way Olivia's sitting, so I'm not sure she's even conscious. Joking aside, is everybody with me? Because there's a lot of information here. Okay, next up, we've got the agranulocytes. The first group of these are probably what you think of when you think in terms of white blood cells. Inside the bloodstream, the proper term is monocyte. Monocyte. Outside of the bloodstream, the proper term is macrophage. Macrophage. Big eater. The main function of these is patrolling the bloodstream, patrolling the tissues of the body. They circulate, and when they come into contact with any pathogen, they engulf it and destroy it. Bacteria, virus, fungus, protozoa, whatever, it is going to engulf it. In the process, they will also release some cytokines to alert the immune system. And then they will do a function called antigen presentation. Antigen presentation. Has anybody here ever heard of this? Good thing we're talking about it. They break down the pathogen. Then they take its proteins and stick them on the surface of the monocyte. The reason they do that is to initiate what we call the adaptive immune response. The adaptive immune response. This is long-term specific immunity. This is what gives you immunity through a previous infection or vaccination. So when you receive a vaccine, very often it is made up of two things, one of two things. Either it is made up of the specific organism you're vaccinating against, either in a weakened form or a dead form, or it is made up of specific proteins and particles from the pathogen. So when the monocyte comes into contact with the virus, let's say, just pick a virus at random, it's going to engulf that virus through phagocytosis. And as you know, that's just basically where it's like an amoeba, it takes it in. Now, once it's inside, it is going to use enzymes from the lysosomes to break that down. But these enzymes will not break it down completely. It will leave specific viral proteins intact. It will then transport those proteins to its own plasma membrane and poke them out. This is going to greatly increase the number of viral antigens in the bloodstream, or in the local tissue if it's not the bloodstream. Because remember, once the macrophage, they're out in the tissue, the macrophages do the same thing. This is going to increase the production of antibodies. That in turn enhances the immune response. Is everybody cool with that? And these are probably the most numerous, and certainly are the largest. They can be up to, your book says, anywhere between 3 to 9% of all white blood cells in a sample. They are much longer lived than the granulocytes. These things hang around for weeks or months. And finally, the last group are called lymphocytes. Lymphocytes. These are the smallest of the white blood cells and the most numerous. Anywhere from a quarter to a third of all of the white blood cells in your bloodstream are of this type. These are the ones that give you adaptive and long-term immunity. Adaptive and long-term immunity. They are further subdivided into two different classes: T cells and B cells. T cells and B cells. T cells directly attack pathogens. There are three different types of them. The first are cytotoxic. And this is not in your book. Cyto means cell, toxin means poison. Shortened form for it are CD8 plus cells. These directly kill infected cells. So if you've got a cell that's been infected by a virus, this thing bumps into it, it will actually kill the host cell to destroy the virus. They also are responsible for killing cancer cells. Then you have helper T cells. These are sometimes called CD4 plus cells. CD4 plus cells. They in turn regulate the immune response. These are the ones that will release cytokines to kick it up, to target specific cells, that sort of thing. The main thing I want you to understand there is it's an up-regulation, Who beeped at me?
That was me.
Don't you beep at me. And finally, you've got Treg cells, which are regulator T cells. Treg cells, regulator T cells. These are ones that are the down regulators. These are especially important in anaphylaxis, and autoimmune disease. Any questions about that? And finally, the last class of lymphocytes are called B cells. B cells. The main function of B cells is to recognize antigens that don't belong and to produce antibodies against them. Once they start producing antibodies, they are called plasma cells. Now, what is an antibody? I'd be willing to bet you couldn't tell me. You've heard about it all your life and you don't know what it is. Okay, it is typically a protein that neutralizes a pathogen, marks it for destruction, and activates other parts of the immune system. It recognizes the antigen, it marks it for destruction, and it activates other parts of the immune system. And all of this is part of the adaptive immune response. So the T cells are the killer cells, the B cells are the support. They're the ones that produce the antibodies. And one last little blurb without getting too far off in the weeds here. Both T and B cells, so let's just say lymphocytes in general, can become what we call memory cells. Memory cells. This doesn't have anything to do with the formation of memories in the brain. What it does is it gives your immune system a permanent record of every pathogen it has ever encountered. And that, boys and girls, is what we call immunity. Six years ago, not a person in this room had an immune system that was primed against coronavirus simply because it had never been out in the wild before. Now, I would be willing to bet probably every single one of us have at a minimum been exposed to it and acquired some partial immunity, either through getting an active infection, getting a vaccination, or simply being exposed to it and not even realizing we have. Is everybody clear on that? Whenever we talk about something like HIV positive, we are referring to the fact that the immune system, number one, has been exposed to the pathogen, and number two, is making antibodies against it. So make sure you get that down. Anytime you're antigen positive, it means your body has been exposed to that antigen. It doesn't tell you anything about how you got it and what dose you got it or when you got it, just that you have been exposed. The second thing, your body is actively producing antibodies against that specific antigen. The general term for that is immunity, acquired immunity. Does my acquired immunity affect Morgan here, since she's right next to me?
Well, I mean, if you're talking in terms of like a herd immunity.
Herd immunity, very good. Does anybody know what herd immunity is? Is anybody conscious? Herd immunity is when a majority of possible hosts have acquired immunity. Why is it significant?
Because it lessens the chances of people who aren't immune to get it because everybody else is immune.
Exactly. There are currently 10 of us in here, okay? Let's say that I am immune compromised. I cannot get a vaccination. And only 3 of you have a vaccination against a particular infection. Does that increase my chances of getting it, Olivia? Does that increase my chances? Let's say only 3 of you have the vaccination. Does that make it more likely that I'll get it or less likely?
More because it's less than half.
Yeah. Because only a third of you have it, right? If 9 of you have it and I'm the only one, well, it doesn't matter if Morgan sneezes on me, does it? Shame on you. Because she is not going to get infected, so I'm not going to get infected. You're not going to infect her. She can't infect me. You can't bring it in. Does everybody see the importance of this? And I cannot emphasize this enough. Herd immunity is a major, major public health issue. I am not going to get out here and beat the drum and tell you you get to be vaccinated or you shouldn't get vaccinated or anything like that. But I hope you understand the mechanism of both acquired immunity and herd immunity. when I get vaccinated for things like the flu, I am not overly worried about myself. Now, I'll joke with you guys about being old, right? But the truth of the matter is, I'm in pretty good health. I've got a pretty good immune system. My mother, however, who I live with, is 84. Now, if one of you did bring something in and sneeze on me, I don't want to take it home to her. So it's kind of a mini herd immunity, isn't it? But I'm able to acquire it where she may not. So that's just something to consider. And I'm going to bring up one last thing and then we'll take a break. Don't worry. I want somebody to raise their hand and tell me what is the single most effective means of preventing infectious disease from spreading, and not you two. Abby?
Yeah. Cleanliness, I don't know.
Very good. Literally washing your hands, practicing good hygiene is the single most effective thing you can do. Take 5. I know. Okay, is everybody seriously on board the bus? Okay, what we're going to talk about next is the last of the formed elements, and it's platelets. Platelets, your book classifies them as cell fragments. I don't really like that term because they are cells without a nucleus, that's true. But they do have the rest of the organelles and cellular equipment. And when you get down to it, erythrocytes are the same way, aren't they? They don't have a nucleus, but they do have an endoplasmic reticulum. They do have mitochondria. And platelets originate from some of the biggest cells in the body. These are called megakaryocytes. Megakaryocytes. And just like the white blood cells and red blood cells, they arise from the bone marrow. Mega means big, karyocyte means big nucleus cell. What these things do is they throw out a blob of cytoplasm and organelles, and then they pinch it off. And that blob is released into the bloodstream, where we call it a platelet. The hormone thrombopoietin is what regulates this. It's generally about half the size of a red blood cell or even smaller. 2 things that differentiate it besides size. Number 1, it's got a shorter life than red blood cell. It only averages about 10 days. Number 2, unlike red blood cells, it's actually capable of amoeboid movement, meaning it can move on its own. And I think everybody knows the biggest and most important function of platelets. What is it?
Clotting.
Clotting. Clotting is generally seen as a good thing. Why? Because of volumetric shock. We talked about that, right? You lose so much blood, you just don't have the fluid to keep circulating. So you want to make sure that you plug the hole. But blood clots can also be a significant danger. When you get a blood clot that forms within the circulatory system and just breaks off, where does it go?
The heart and the lungs.
It can go to the heart and block one of the arteries supplying the cardiac muscle. We call that a myocardial infarct, a myocardial infarction. Myo means muscle, cardio means the heart muscle infarct means, oh, you just had a heart attack. Once the cardiac muscle dies, do you get it back? No.
No.
So if you lose 20% of your heart function, congratulations. For the rest of your life, you're now at 80% max. See why it's a worry? Anybody here ever heard of a stroke? An ischemic event? You get a blood clot blocking one of the vessels in the brain. All of the tissue downstream from that clot dies. Can it have a significant impact? Absolutely. Blood clots in the lungs. You get an idea here? So it's very important that this process works and is regulated. And platelets play a key role in that. The way they do it, they're bouncing around and circulating throughout the bloodstream. Normally, they bounce off of the smooth inner wall of a blood vessel. Does that inner wall of the blood vessel stay smooth once it gets cut? It rips it apart, doesn't it? And those jagged edges and ends catch up the platelets. When that happens, they actually break apart. This causes the interior of the platelet to secrete several chemicals. This process is called activation. These chemicals are now circulating in the blood. They attract other platelets to the damaged site. This is called positive chemotaxis. Positive chemotaxis. Those other platelets also break open and they attract even more. You see how this is going, right? It's a cascade effect.
Positive chemotactis?
Taxis, T-A-X-I-S. Are you saying I sound like I'm from Tennessee? Boy, howdy.
How about them balls?
Roll Tide. Anyway, they also react with a soluble blood protein called fibrinogen. Fibrinogen. This converts the fibrinogen to a non-soluble protein called fibrin. Fibrin. Since it's no longer soluble, it's going to fall out of solution, isn't it? It's going to precipitate. It's going to stick to that mass of platelets and jagged edges and start forming a net, almost like a spider web. At the same time, the blood vessel is undergoing something called a vascular spasm. A vascular spasm. It constricts. It chokes down. Why is it going to do that? To minimize the flow of blood through the damaged area, right? That's going to slow the bleeding. So now we've got platelets hitting this network. They're breaking apart. You've got more fibrinogen being converted to fibrin and falling out of solution. You got this big massive ball of sticky stuff forming. Red blood cells begin to adhere to it and plug up the hole. Eventually, there is enough debris and cells, and protein, and just junk caught in that gap to close it up completely. We have formed a plug, and that plug is called a clot. What is the difference between a clot and a scab?
The scab would be hardened.
Bingo. The clot, when it first forms, almost has a consistency of jelly because there's still a very high moisture content in there. It is the immediate plug that stops the bleeding. Is everyone clear on that? Clotting is only a process of minutes. Five to 15 minutes is typical. Now that depends on a lot of factors like what you have in your blood, your overall health, blah, blah, blah. But has everybody got the idea? It's quick. It has to be, because otherwise you're going to go into volumetric shock because you lose, or hypovolemic shock because you lose so much. Now, once that jelly-like plug is in there, it's going to begin to desiccate or dry out. That's normal. Eventually, it's going to form a dried mass. We call that a scab. Now this is living tissue, so underneath that scab, you have tissue repair and regrowth occurring, don't you? So the scab acts as both a plug long-term and as a protective shield or layer, keeping the new tissue from pulling apart, being further damaged, and so forth. And eventually, it will fall off, and you'll be left with a healed scar. So the clot is the initial clump. The scab is the long-term cover. Everybody cool with that? And I'm not really going to get into it beyond that. Everybody understands what platelets are. Typically between 150,000 to 400,000 of them per microliter. If you have thrombocytopenia, that means low platelet count. Means you're more likely to bleed. Thrombocytosis, high platelet count, that can cause clots to form. Do y'all want another break now? Nod, drool, or otherwise indicate consciousness. Okay, we'll take another break.